לא באמת להתמודד מאותו מרחק, מתוכו ומתוכו.
יש לי, ברור שאם אני אסתכל עכשיו על הדאטה סט שלי, ואני אקח את כל התאים הפרמגליים שבדקתי מהם, והם יהיו אותו טייפים, אבל לא יהיו איתם אותם טייפים, זה לא יהיה כאילו שמדד אותם אותו.
הקוד שיש לך פה, אם יש לך טרייס כזה שיש פייל, ואז לראות דברים כאלה, הוא יספור בזה.
כן, אם זה מצב מפרש שלו, כן, בגלל זה, אחר כך כשהיינו עושים סייט-סורטינג, אז היינו רואים כאילו צורה כזאת, אבל היינו רואים משהו כזה. והיינו רואים את זה הרבה פעמים, כאילו כל אחד מהם. היינו רואים שזה נראה שלא.
יש לי גם HDMI. יש לי, לא, יש לי HDMI. למה זה יותר טוב?
בוא נראה אם זה עושה. with the HDMI זה אוטומטי.
זה ישר, זה בכוח. יש פה יתרון.
עכשיו אני מבין מה היתרון.
מה? לא, אתה לא.
כן, כן.
זה היה קורה גם ב-HDMI?
כן, כן, זה לא בזה אותו דבר. אני גם, יש גם עוד משהו.
it's okay. Sorry, this is in English, right? Good afternoon. And welcome back after a short break. I understand you're now experts in Yuval Reader. Kind of. Okay, good. So one of the take-home messages of my part of the course is that Yuval Reader are entirely wrong. Giants, definitely. They changed the field of vision research entirely and set it back by about a century. So we'll discuss that. We'll get to know the real of me. This part of the course is put by myself and Udi Zahari, and he's going to do the student lectures. And some of you got already papers for that? Okay. And we're going to do 3 sessions, and then we have a break for 2 sessions because of Yom Ha'atzmaut and something else you're doing, and then we'll have a 4th session, and then Udi will come and do a week later a session. Okay? Did you receive a syllabus and things like that? Yeah? Okay. And the reading, you have the text? Yeah? Okay. So I don't have. What? I'll upload the presentation after the lecture. Okay. The principle is, the claim was that the definition of a university lecture is that material passes from the notebook of the teacher to the notebook of the student without going via the ear of their head. To try and avoid that happening, I would like this to be more of a discussion and things happening, and so I'll send it afterwards and then you'll have to know. But I will. Okay, so what I would like to do today is to talk about 7 theories of vision. This is the kind of introduction to the next 3 sessions. Okay? Because we want to know what vision is about. What does it mean to have perception? Okay? And most of what I'll say about vision is true about the other senses as well. But we'll talk about vision because that's what I'm supposed to be teaching. Okay. So let's start with a simple one, which is Kepler-Hartline. Kepler-Hartline did 2 important things. He defined the receptive field, which you've learned about, and he recorded from a single neuron. And the story goes like this. He spent his summers in Woods Hole. Woods Hole is on Cape Cod off Massachusetts. Why is it important? Because it sticks out into the ocean, and they're able to catch all kinds of fish and other sea animals which actually live in the middle of the United States. Just like Plymouth in England, which is where they did the most important experiments on squid, okay? Where they have a marine biological lab. But it's also sticking out into the ocean on the other side. Okay? So it's very important to be able to get the right preparation. So one summer he spent at Woods Hole, and he just told the supply department, catch anything you can, give it to me, and he studied the eyes. And he would shine light on the eyes of the fish or the crabs or whatever he could find, and to get the responses. And one that interested him especially was this limulus, the horseshoe crab. And he liked it because it has lots of eyes. It has 2 lateral eyes and 2 medial eyes, and has some eyes underneath, and there's 1 eye in the middle of its intestine, and so on. And so he was able to shine light and to see. Okay. And this is not a simple eye like we have, but a compound eye like a slide. In other words, many omonotia, so he could easily shine light on one of the eyelids. Okay? And then he found that if you take a hacksaw and you saw the shell of the crab, and then you carefully take out the thing and clean off the nerve that goes from the eye to the brain, if you want to call it that, then you can eventually cut off the end, put it into a dish of seawater, and you can put the nerve onto a little hook, and you can record He said, well, that's nice. Now, next time what I'm going to try and do is separate this optic nerve and get one fiber and be able to record from one of the fibers that comes from the eye. And then I'll have a single cell. I'll know how a single cell responds. And I'll be able to shine light at different parts of the eye and see how it responds. Well, he spent all summer and how does he know if he's recording from one cell or two or five or whatever? By the shape of the spike. Actually, he didn't really look at the shape, just the size of the spike. Some are larger, some are smaller. Okay. So he got 2 or 3 or 5. He couldn't get one. Last day of summer comes. He goes to the supply department to do his last experiment, and there's this one large, old limulus, and they're hardly moving around at all in the tank. He says, that one's too old to use. I'll go and play tennis. But he's hard-lined. He doesn't do that. He takes the old one and he records from it, and he gets his single neuron response. He realized eventually that the reason that he was able to get it was because most of the cells were dead. So even if he had 5 fibers, he was only recording from 1. But that's how it goes. Now, why are these two things related? The receptive field and the single neuron. Because you define receptive field in terms of a single neuron. What is the contribution of that one neuron to vision? Okay? I did my postdoc at Israel. Where he told the story. Okay, so the definition is the area of the world around the retina where change in the stimulus induces change in response in the recorded neuron. And that can be a neuron which is in the eye or in the brain or anywhere. Okay? In the spinal cord. The receptive field of the spinal cord. Good. And usually it's done, let's say how Euler and Weasel did it, was to have a cat sitting on a table, usually anesthetized. So his head is usually in some sort of clamp. And he's looking at a TV screen, or what they were using was just a board, and with a electrode into a hole cut into the skull. And why doesn't the cat feel anything, by the way? Why doesn't it hurt the cat?
There's no pain receptors on the brain.
Right, okay. So there are no pain receptors around, and therefore even if this cat was alert, he would not be feeling pain when you put the electrode into the in this case, of course, the cat is anesthetized. Okay, but with monkeys, they're not. How do we know that there are no pain receptors in the brain and that therefore he's not feeling pain?
Know how pain receptors look like? You can find any?
Okay, you can't find it. But just to be sure, what's another way of making sure that the monkey is not feeling any pain when we put the electrode in it?
Behavioral.
Behavioral. If you squeeze his finger, he screams, and when you put the electrode in, he doesn't. He doesn't feel it. He doesn't know that it's there. And the same with people, by the way. They don't know that it's there. The same thing is done with people. Okay. Now we have to extend this definition of receptive field ultimately, because in areas where you have face cells, you've heard of these already, that there are cells which respond to a grandmother. We call them grandmother cells. A grandmother cell is a cell that responds to my grandmother, whether I see her here or there, in large or in small, and wearing any different clothing and so on. I know that it's my grandmother because there's a cell in my brain that responds to the grandmother. Is that really true? Well, maybe. But if there is such a cell and we want to define a receptive field, what is the receptive field of the grandmother cell? The whole visual field, okay? And the one that responds to a table. The whole visual field. So what, is there no difference in the receptive field? Hmm? There is. So what's the difference?
Space isn't the retinotopical space. Space is the abstract space of ideas.
Exactly. So one has to extend the notion of receptive field from space in XY coordinates to space in multidimensional coordinates, which includes objects and so on. Okay? So the meaning of receptive field has to change in that way. Okay. There's another thing that's important about receptive field. It teaches something about the connectivity of units. For see this? Yeah, if I turn the light maybe. Yeah. So, if we have a cell which has a receptive field like this, and another one which has a receptive field like this, and another one which has a receptive field like this, and then, that's the only color, well, we have black. And then we have a cell which has the receptive field like this. Maybe we can infer that this cell is summing those. Okay? And if this cell responds to a stimulus that's like this, but if we make it longer, it does not respond, what does that mean? You didn't get this from Yuval and Weasel. He had something called end stopping. No? No. Okay. What Yuval and Weasel found was that there are simple cells. They first found it in complex cells and they called them hypercomplex. But then they found it in simple cells and they threw out the notion of hypercomplex. I'm getting a little bit ahead of myself, but it's worth it. And they said that against the length of the bar, the response is sometimes like this, but sometimes it's like this. What does that mean?
We have inhibition on the cells.
Exactly. There's inhibition. I wish I had more color. Sorry. So it seems there's another cell here which gives minus inhibition to these give plus. So these are summing and these are inhibiting. Okay? Any other models of how this could work?
This is the model that they suggested, right?
What?
This is the model they suggested.
This is what they suggested, yeah. They called it end stopping. Some cells have end stopping, some didn't.
So suggest another model.
Now I want you to suggest another model. I just had a model where I have 7 LGN cells which are feeding into this one simple cell, right?
Lateral inhibition in the cortex.
Lateral inhibition in the cortex. So what you're saying is we can have one cell which sums these three, and another one which sums these three. Well, but there's mutual, and maybe another one here. Right? But these two inhibit that one. Clear? Yeah? Now that we have two models, the question is, yes, go ahead. This model says that the LGN cells inhibit. This one says they're cortical cells which inhibit. How can we tell the difference between these two? At least the way I view it.
They can't be retinal cells.
Hmm?
They can't be retinal cells also.
They can be retinal cells also. What do you mean?
Because you have inhibition from cells in the, like from photoreceptors in cells.
Yes, I mean, I said this was LGN because it's the LGN that feeds into the V1 rather than the retina.
Right, so I'm saying, can't you also have lateral inhibition in the retina itself?
Let's say you could, but you're giving a third model. I want to know now, how would you differentiate between these two models?
Cut the inputs from the LGN.
Well, but then you won't get this one either.
You can activate both neurons with their receptive field on the right motor. You can activate two of these neurons and see if there's mutual negation between them.
Okay, say it again. You activate.
You activate, for
this.
example, the bottom and the middle neuron
How do you activate it?
with the electrophysiology, current
Okay.
injection.
Okay, so you want to
Also genetic.
put an electrode in, activate the cell, and see if it directly does it. Very good, yeah, very good way of doing it. You're an electrophysiologist, that's how you would test it. You would put electrodes into this cell, and this cell, and see if there's inhibition between them. Okay. There's another difference between these. Okay. In this model, when does this cell respond? It responds when all three of these respond, right? To a line. What happens if we put a stimulus which is not a line, but a line this way?
Probably won't have enough to respond.
It would not respond. Okay? But this cell would respond, and this LGN cell will respond, and it should inhibit. In fact, if we put a few of these lines, this cell will not respond because the orientation is in the wrong direction, but these will respond. So they should inhibit. So we can differentiate between cortical inhibition and direct LGN inhibition by whether the inhibition is oriented. Okay? Got it?
I'm not following. How do you know that those cells are selected for horizontal stimuli?
No, they're not. The LGN cells don't have orientation dependence. They respond to any direction. That's why when put this, they also respond. But if it's the LGN that's responding, then it doesn't matter what the orientation is. Okay, go ahead.
So if you give, just to understand what you're doing, so if you give a horizontal stimulus, so you expect the inhibitory cells to respond,
Right.
and not the excitatory cells to respond.
I expect the LGN cells to respond, but not the cortical cell. So if the inhibition comes directly from the LGN, it will inhibit. If it comes via a cortical cell, it will not inhibit.
But if you're presenting horizontal stimuli, there's nothing to inhibit.
I'm sorry. The stimulus that I'm trying to present is a stimulus like this,
Yeah.
which excites that, and then in addition to it, instead of a continuation, lines like this. Okay? Okay. And not only is it true that end stopping depends on the orientation, but a line like this will not inhibit. Okay? Because it's got the wrong orientation down here. Therefore, these end-stop cells become curvature detectors. They respond only if the line is straight and not if it's curved. I'm sorry. They do not respond when it's straight, and they do when it's curved. Okay? They're inhibited by this and not by this. Okay?
So which model is true?
The model of that it's cortical is true.
And there's no LGN inhibition to the visual cortex?
I didn't say there's no. I said the end stopping is not caused by it.
Then,
I'm talking about end stopping. Okay?
so
The reason that I point this out is, first of all, you got to test things and think about them. Second, here is a detector for curvature. And it comes about in a very simple way by having end stopping.
what kind of inhibition does come from the LGN?
We're going to go back again to the one and get to it. But the dependent, for example, in the V1, there is responses not only to bars of light, but also to edges. In order to have a response to an edge, you want to have an emission from here and excitation from here. You want to have two rows like that. Okay? So that's an emission and excitation, one next to the other. A simple cell which responds to an edge rather than to a line. Okay? Good. Okay. So, the next one we wanted to talk about was Euclid and Weasel, and we've already said a lot about Euclid and Weasel. So there are a number of things which are important about Euclid and Weasel. Cytoarchitecture, finding that there's organization within the area. Okay? There are columns and hypercolumns. This you all know about, right? Columns for orientation and for, that's all important. Number two, and maybe the most important thing that they did was to say there's a hierarchy of integrating and generalizing units and areas. Why do I say integrating? Because you have many LGN making a simple, many simple making a complex. And at first they thought there are many complex making a hypercomplex. There are many hypercomplex going on until we get to the grandmother cell. That's the model they never really wrote, but certainly talked about, and they implied, and everybody heard. All we have to do is sum things together and we get the right answer. Why do I say generalizing? I think it's an important term. Generalizing. What do I mean by generalizing? In what way does a complex cell generalize over simple cells? A variant of position. It always responds to a bar, always to a thin bar, not to a thick bar, but invariant over position. Okay. Good. Here they are at work, and this is the way they used to work. They had a slide projector, which used to be these huge, massive things, in which you put a slide. The slide, you guys never even seen slides, right? Yeah, you know, that big, and they have a glass piece on each side. And you've heard the story of the slides of Euclid and Weasel? Okay. So they had the slide, and they had lots of slides with a black dot in the middle, which is small and bigger and bigger and bigger, and they had lines, and they had all black with just a white hole in the middle. And one day they had one of these slides with a small dot in the middle, and it fell on the floor, and the slide cracked. So they were going to throw it out and make a new one, but they couldn't wait because they were in the middle of doing the experiment. And here's the cat sitting on the table looking at the screen, and they're marking the receptive field very carefully. How do they mark the receptive field? With a marker on this page, okay, on a piece of paper, right? And how do they know where the receptive field is? They listen, exactly. They listen to the speaker. The speaker is attached to the amplifier. It amplifies the noise of the spike. So when they put a line in the right orientation, in the right spot, they go psh, psh, psh. When they turn it, nothing. Okay. How many cells were they recording from? 200. More than one. They certainly didn't know. They weren't looking at the oscilloscope. All they were doing is shh, shh. Okay? So they probably had lots of cells, and that's probably why everything was so nice and neat, because they were averaging over cells. So all these cells had an average orientation preference of this, and then when they moved along, they slightly changed because the average slightly changed. Did it change from one cell to another? Who knows? In a way, it helped them because they were able to see the organization. But it wasn't accurate. They weren't trying to be. So the slide fell down, and they put the slide back in, and this time the cell responded vigorously, more than they ever had heard a cell before. because the cells don't respond to points of light, they respond to bars. And this crack in the slide, very faint, was a bar in the right orientation. And that's how they hit upon the notion. Okay?
Good.
Right? Say it again?
It's true.
Is it true, the story? Sure it's true. Why do you think it's not true?
Okay, I know. Nice story.
They told me the story. They told the story of lots of, they like to tell. Why do they like it? Why do scientists like to tell this kind of story? It's obvious that I want to make the point. Because the greatest findings are made by chance. Okay? By something happening, okay? By parrot having this monkey there, and he's doing all kinds of stimuli, and then he walks between the screen and the monkey, and the cell starts firing a lot, and he found the red cell. That's how he found it, just by chance. And he could have just forgotten, well, that's curious, doesn't matter, it's so fire. Okay, well, maybe the line there. No, okay. So you have to look after these things. Okay. This is the only graph that Euler and Wiesel ever published. The only graph. Nothing else. It's all words. And this graph says how the orientation changes with position of the electron when it goes across. This, they did not publish, but anyway, this is how left, right changes as they go across the field. This is not there, but it's what they found. And you have left and you have right, and in between you have right but a bit left but a bit right, and you have something in between. So binocular cells are between the ocular dominant cells. This you did remember. And you know the icebox. I thought you studied human weight. What paper? Which paper did you study? The Ferrier lecture. Yeah? Okay, what's it? Anyway, the icebox picture, their picture, shows that it's left, right, and orientation horizontal, vertical, going horizontal. And if you take this area of cortex, that's called the hypercolumn. Each one of these is a column where all the cells have the same orientation and the same ocular dominance. Altogether, this gives you, okay? Now there's a problem. There's also a map of the world across the cortex, right? So that this point is there, and this point is there, and this point is there, right? That means that at this point in the world, you can only see vertical. And at this point in the world, you can only see horizontal.
We have more columns.
Yes.
More columns.
Yeah.
That make up the.
So explain.
In other words, you have one column where you have vertical at that point, and then you have another column where you have horizontal at that point.
So all of these have to be the same point.
Yes.
What you said earlier is relevant only to the left.
No. There is a map of the world on V1.
Yeah, but it doesn't. There is a map of the world, but it's like an area map. There is an area that responds to a point, and then that's another area that responds to a point.
Okay, so all of this responds to one point, and then all of this responds to another point. Okay? How many points on the retina?
That's what you see.
Million?
Huh? How many columns would you need here?
But the apple responds to a point. I don't see it. Right?
Yes, that's true. So it responds to a field like this, and this one responds to a field like this. The answer is this jitter. If you go from cell to cell, they're not all exactly in the same position. So when you go from here to here, you move one receptive field width. But along the way, you have all kinds of different positions, so that somehow you do cover the entire area of the receptive field. Okay, let's move to theory number 3, because otherwise we're never going to get anywhere. Theory number 3 is David Marr. You've also heard of David Marr, right? David Marr lived to the age of 35. A bit less than Mozart. He is the one who started artificial intelligence of the visual children. Okay? He started out in other stuff. He then came to MIT. He met the artificial intelligence group there, Marvin Minsky and so on, and moved into that field and discovered that computer vision is very hard. That was the most important thing he found, that the computations that are needed are not simple. They're very difficult. And they still haven't been worked out, despite everything you're hearing. Okay. And he said, first of all, he called his book Vision. The book came out after he died. It came out in '82, and it's been reprinted now by a bunch of his students, including Shimon Uman. Vision, a computational investigation into the human representation and processing of visual information. It's very important that he talks about both representation and processing. There are two things that are going on. First of all, we're creating a representation, and how are we doing it? Via processing. So you have to process the information and get a representation. Okay. So you need to study both. And he talks also of the hierarchy of feed-forward computations. He accepts Hubel and Wiesel hook, line, and sinker. Okay. He really talks about feed-forward processing. It's one of the big lacks. There's almost no feedback going on in this whole book. Vision is difficult, but it's a solved problem. You've heard of that term? Yeah? That means we know that vision is hard, but it's possible. How do we know it's possible? Because we have it. Since we have it, therefore it's solvable. Now we can find how to solve it in the computer. And we can learn a lot from biology. Biology has solved the problem already, so now we can learn how biology did it and maybe learn some tricks from there. Not necessarily that the best way of doing it by computer is by doing it the same way biology does it, but at least we know that it's possible. Complex computations being performed by simple algorithms, we'll get to it next time. What does it mean to see? Now here's a quote from, and you've seen part of this quote at least. What does it mean to see? The plain man's answer, and Aristotle's too, would be to know what is where by looking. In other words, vision is the process of discovering from images what is present in the world and where it is. Okay. I hope that by the time we finish this course, you will say this is ridiculous to say that vision is to know what is where by looking. Okay. It's a much more complex one. He talks about 3 levels of investigation. Did you talk about this stuff before? Yes. Okay, good. So I can do it quickly. Computational theory. What is the goal of computation? Why is it appropriate? You have to talk about the goal. If you don't know the goal, you can't know what's going on. Representation algorithm. How do you get to that goal? What is the computation you have to do? What is the algorithm for finding it? Okay? If you want to know that there's motion, motion means to change its position over time. Not true, but for the moment, let's say it's true. To change it, so in order to know motion, you want to know if something's moving. So just look at it here now. Look at it there now. Know the position, and take the difference in position, how long it took to go from here to there, then you know the speed of the motion, right? That's the algorithm. Now, how do you do that? Okay. Well, you take a light sensor here and a light sensor there, and it'll open the door when the guy moves faster. Or, okay, so biologically, you might do it differently than you do it in a computer. So, there's, first of all, the goal. The goal was to know motion. What is the algorithm for knowing it? Know what's here, know what's there, know the distance. Divide delta x over delta t. Hardware information, how can it be represented and the algorithm realized physically, in biologically or in a computer? Then he's also got 4 levels of representation. Did you do this too? No. Okay. 4 levels of representation. First is the image. Where is the image? It's a representation of the intensities of light at different positions. That's on the retina. Those are the photoreceptors. The photoreceptors know exactly how much intensity of light there is in each position. There are 3 types of cones. We might know also the color. Okay? Intensity value at each point on the image. The The next thing is a primal sketch. There's some sort of sketch of what's going on. What David Marr is saying is you have to go through these 4 steps in order to know what there is in the world. Knowing what is where happens not in 1 step, but in 4 computational steps. The first is getting the image. The second is to make a primal sketch. Make explicit important information about the image, primarily intensity changes and geometrical distribution and organization. What is the organization of these lights and darks? Okay? If I tell you that on this screen it's white, 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 blue, 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 white, 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 blue, white, 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 blue, white. That's how fax works, by the way. Okay? How does it, do you know that? That's how fax works? The fax sends information back, 10 white, 2 blue, 5 white, 1 blue. That's how fax works. It's much shorter because it's just, okay, and if you put the paper in straight, then you save a lot of, if you put it crooked, then it takes longer. Okay, so you got to know not about how much where there's white and black, because that doesn't tell you anything about the thing. But you have to know at least where the changes are. Okay? So you use zero crossing. A zero crossing. We'll get there. Anyway, it's a way of finding an edge. So you have to find edges. You have to find blobs. There's a lot of blue here and a lot of white there. Termination. Endings of lines. Discontinuity. X segments. Virtual line segments. Third level is what he calls the 2.5D stage. It's somewhere between a 2D and a 3D sketch of the world. Okay? The last one is going to be the full 3D. You make explicit the orientation and rough depth of the visible surfaces. The contours of discontinuities and the quantities in the viewer's tended coordinate frame. I'm looking, so I know there's an edge there and an edge there. Okay? Local surface orientation, distance from the viewer, and so on. And the 3D model will be a description of the shapes of the things that are out there in the world. And now it's going to be in 3D, and it's going to be with the depth, and each object is going to be in terms of the object itself. The object is small here and large there. So the 3D model is arranged hierarchically, each one based on the spatial configuration. Volumetric and shape primitives are attached. Okay, that's David Marr. We're going to do one more and then we'll make a break. Because this one is a total break from this whole notion of hierarchy, receptive fields, and things like that. Instead, comes along Jerry Lepton with his famous paper called What the Frog's Eye Tells the Frog's Brain. And what Jerry Lepton said was, what's important is, what is the goal? Like Marr asked, what is the goal? But let's look at the goal when we're studying the retina of the frog. Because the frog's retina should not tell the brain just that there's light here and there and there and it's dark there and there. That's not important. Okay? And he found cells in the retina of the frog that respond to a dark point, dark meaning darker than the background, and is moving quickly in any direction. So this cell responds to something moving over here, and the cell next to it responds to something moving over here. And the cell next to it, something moving over here. And what's the response of the frog? The frog's response to this cell firing when there's something moving around here which is dark is to stick out its tongue and to grab it. Because it's probably a fly. It's a bug detector. And there are 3 other types of cells that they found. One was general dimming detectors. If the whole thing dims, what does that mean? Why does the whole scene dim?
Strong light, though.
Maybe there's a strong light outside, so why did it dim?
Sun is coming out.
To not waste energy when it's not needed.
It's
To notice shadows.
Say again? It got dark. Why did it get dark? coming, there's a shadow. And that thing that's coming might be a predator about to eat the frog. So what does he do? He jumps off the lily pond into the pond. And that's what he does when there's a shadow. Then there's the third kind of cell. The third type of cell sees everything in the scene moving. Whole scene moves. This way or this way. Everything, the whole scene moves. Why does that happen?
Someone is holding, or maybe upside down.
Okay, he's sitting on the lily pad and it's shaking in the water. Okay? So what does he do?
Balances it by
What do you do when you move your head? Look at the thing again, now move your head.
move your eyes. Move my eyes.
You move your eyes in the other direction. So that's what he does. When the world moves, he stabilizes the image by moving his eyes. So he's got a way of getting his food, a way of keeping his image stabilized so that he can see that bug moving, and he has a way of avoiding the predators. All in the retina, directly perceived in the retina. One last thing before we do make a break, right? So we said already that when you move your head like this, so now move your head pretty fast back and forth, and you see that finger pretty go out, right? Now, instead of moving your head, move the finger at about the same speed. It's pretty blurred, right? Why is it blurred this way and not this way?
When I move my head, it's always in the center of my retina, and when I move the finger, my eyes stay still and it goes to the side of my
Why
retina.
does your eye stay still? Why don't you move your eye?
It's not that the whole world is moving, just the finger is moving.
Okay, we'll put a white screen behind it so that you don't see anything but the finger. Now we'll do the same experiment. In this one there's a lag, and in this one there's no lag. Why?
Because you have the representation of the movement.
The neurons that move my eye, know where my head is.
Okay, so that's one answer. The neurons that move, yeah, but the neurons that move the finger don't know.
The brain.
The neurons that move the finger were also the brain. Why didn't they tell the eyes to move?
Because we want to see things moving.
We have crystals under your
What tells us that the head is moving? We close the eyes. Close the eyes and move your head. Vestibular. The vestibula controls the eye movement. And the vestibula has three orientations, right? Three semicircular canals. In which directions are they? They're exactly in the same directions as the muscles of the eye. Lateral, up and down, and around. So they're set for moving the eye. And therefore they respond very, very quickly. As soon as the head moves, the eyes move in the opposite direction. So we're doing exactly what the frog is doing. What the frog is doing, not with his vestibula, but he's doing it with the retina. Okay? And he's doing it very well, keeping the image stabilized. It's a very important part of vision to keep the image stabilized.
Doesn't use it zero.
I don't know.
How can you use the retina to keep the retina?
He's using the retina to keep the retina because he's keeping the image stabilized.
But how does he know where it moves?
He
know
sees
what-
the entire image moving and he moves his eyes to keep it stabilized.
By reference. So
Yeah.
Like optical image development.
Right.
Don't we do this too? I mean, besides the...
I imagine we do it too, even without the vestibular. But the speed of doing it that way is okay? Short break.
באמת, זה בסוף זה הכוונה שלכם. זה stop equation one. איך אתה מודע לפתרון הזה? נעשי פרוגרס, לא מבין מה שזה נראה.
מה, תעשה עוד קונסול, לא?
אז למה אתה צריך את זה? למה אתה צריך אחד? למה שהוא יכתוב אחד?
אוי, זה לא נורא.
שזה שתיים? למה? סבבה, למה לא לשים את זה בסוף כאן?
אז כאילו אין לך פתרון פה. זה נכון?
אז זה לא מה שאני
we have all these LGN cells, which sum up to give one cortical cell, right? And why does it respond to a bar in this orientation and not this orientation?
Is all the cells to work together
The cells should work together.
and not to get inhibition from the cells from the side.
Not to get inhibition from the, okay, good. Now, we have another set of cells which create a neighboring simple cell, right? And how do we make a complex cell?
Several simple cells.
Well, several simple cells. So we're going to add up these two and a whole bunch more, and this is going to be the complex cell. And the complex cell will respond to only a narrow bar, not a broad one, and only in the correct orientation. Okay? So when I give a bar here, I get a response. Let's call it 10 spikes per second. Spikes per second. Okay? And when I give this bar, then I get 10 spikes per second. And when I give them both together, what do I get? 0 spikes per second. So 10 plus 10 is 0, right? That's what they say. Is there a problem here?
Non-linear.
What?
Non-linear.
It's nonlinear. How is it nonlinear?
And plus then supposed to be 20.
You're right, it's nonlinear. You're right. See? At least you already showed that it's not linear. What is it? Does it bother you, Enrico? Not linear. They say, yes, complex cells were more difficult to map their receptor fields. We could not do it with points of light and even simple, etc. But so they're complicated or they're complex. But the model says all they do is add up many simple cells. They never use the term nonlinear. Okay. We'll talk next time about what nonlinear means. Okay? Let's go back. Okay. So what Jerry Ledfin did was say what's important is to look for bug detectors, predator detectors, locomotion detectors, trigger features. And this happens in the retina, and it happens irrespective of the level of illumination. These same cells are responding, whether it's very bright or very dim. They're still going to see that bug, as long as the bug is dark on a brighter background, because that's what bugs are. They give prominence to what is informationally important. And there's an immediate direct detection of some, what we would normally call high-level information, detecting a bug. Okay. Let's go to Gestalt. You've also talked a bit about Gestalt, right? And mainly, I assume you talked about the rules of Gestalt. Things that are linear, you see these points as being vertical rather than horizontal because they're further apart. Yeah? No? So what did you talk about Gestalt? So what is Gestalt? No? What is Gestalt? Hmm?
Talk about the holistic.
Holistic aspect. Okay. So the Gestalt people said that what you see is the holistic view. Okay? In the chapter that you were supposed to read for today, it talks about also some of the features that they talk about. That you see, for example, if I draw two lines, which line is the continuation of which? The drew it well, okay? You don't see it as this, even though I drew it that way. Okay. You see good continuation. Okay? And if you see points like this, are these columns of lines or horizontal bars of lines? Columns. And if I do this now, add a few more columns, what do they become? They become horizontal. Okay? So proximity has, and they have all kinds of rules like this. And that's a lot of what's talked about in the show. But the main thing that they talk about is what you talked about, which is the holism. That you see the whole picture, not just the parts. In fact, they not only say that perception is not constructed from atoms of elementary, unrelated local sensations that are unified to associations through the spatial temporal. It's not that we build up the whole. We get the whole directly. It's the whole that counts. And the whole determines what the parts will be. We know this is an arm because it's attached to a body. If it was floating out there, you might not see it as an arm. The whole is something else than the sum of its parts. Not more than the sum of its parts. It's something else. Because summing is a meaningless procedure. You don't sum the parts. Whereas whole part relationship is meaningful. And Wertheimer said, holism is the fundamental formula of Gestalt psychology. There are wholes, the behavior of which is not determined by that of their individual elements, but where the part processes themselves determined by the intrinsic nature of the whole. Okay. I think I've got one more. Ehrenfeld, who began the whole thing, noted that perceptual experiences, such as the perception of a melody or the shape of a visual object, are more than the mere sum of their independent components. A melody is not just notes put together. That's maybe physically what it is, but that's not what we perceive. We perceive the melody. And after we perceive the melody, we can perceive the parts. And we account for these by postulating a new element called the Gestalt. Now, is that always true? Are all dimensions always part of a whole or not? So there came along a guy by the name of Wendell Garner, worked at Yale University for a long time, died recently. And he said there are two kinds of ways that dimensions can unite. They can be separable or they can be integral. They can count together or not together. Okay? So what is, I don't know which way to start, separable or integral. Separable dimensions. If you want to judge, let's say I'm going to show you either a red triangle or a blue triangle or a red square or a blue square. And I'm going to ask you the shape. And sometimes I'll show you a square which is red and sometimes blue, and sometimes the triangle will be red or blue. Will that change in color interfere and make it slower to say whether this is a square or a triangle? And the answer is no. Those are separable dimensions. You're able to attend to the shape without thinking about the color, and vice versa. But let's say I ask you, is it blue or red or pink or tchelet, light blue, where the differences are in saturation and hue, those are integral dimensions, and it's harder. It's slower to say if something is bluish or reddish when it's sometimes blue and sometimes tchelet and sometimes red, sometimes pink. Is there anybody here who doesn't know Hebrew? At all. Okay? So you now learned one word in Hebrew. Tchelet is light blue. What? Okay. There's no word in English for it. Okay? There's no one word for it. There is in what? Sand, no. There is no word for chet. There is in Russian. And there, okay. No, we've done a study to see how quickly people can actually differentiate. Okay, and it depends on their language, what their mother tongue is. Okay. So it's integral means that what they call garner experience. It's slower to do. Now, what happens if we vary the two together? We always show either a blue triangle or a red square. And we ask you, what is the shape? Is that faster than if we vary? No. If we show you either pink or dark blue, that's faster. Because you use the two things together. You have two hints. Okay? So there's a difference between integral or separable dimensions. Okay. And the same thing happens with attention. Can you attend to one and not to the other? And finally, there's another difference, which is how different are they? If you ask people to judge, how different is this red square from this blue triangle? What they're doing, they just give a number. Give a number 1 to 10. We show various shapes and various colors and so on. And they give a number. The number that they're giving has to do with the sum of the difference of how different is blue and red and how different is triangle and square. Okay? When they're separable. But when they're integral, it's the square root of the sum. It's, instead of being dimension 1, dimension 2, and the difference is the sum. Let's say this is red and this is blue and this is square and this is triangle. Then the difference between this point and this point is like this. But if I'm doing the same thing with blue and red and pale or dark, then the difference is this, the square root of the square root of the sum. Okay? This is what they call city block, and that's the 2D space.
To me, this is a response that you get when you just ask people to give numbers.
Yeah.
I'm not sure why that should be meaningful. In other words, shouldn't that be like dependent on? I think that's more dependent on how people perceive how you want them to give a number as opposed to what their actual perceptual differences. Like, I don't think that that's a good way
It is
to
an amazing thing about psychophysics. People are very consistent, and they're very good at making judgments. If you ask people how loud a noise is, they're very good. They're very consistent. Well, they're what's called the magic number 7. They'll do 7 gradations of loudness. I have 7 gradations, 7 intensity of light, etc. It's always what they call the magic number 7 plus 9. But you can ask people to make judgments, and they're very good at it. When you ask different people, you get the same kind of result. Okay? And it does not depend on that. I mean, you always have to be careful how you're asking the question. So, in all psychophysics tests. But if you've done it carefully, you can depend on people's response.
But can't you cross a category boundary of colors by just changing the hue of brightness without changing the actual wavelength? I mean, people perceive definition of colors. It's not only about the, I don't know, the wavelength of the.
And there's 3 aspects to color. There's wavelength, intensity, and saturation.
Okay. So but people don't actually define in their language this.
They don't. That's correct.
But so I can give a person 2 colors with the same wavelength,
Sure.
but with a different saturation or brightness,
Blue,
and you
like
would
pink and red.
name them. You would name these 2 colors differently.
You name them differently and tell you how different they are.
Yeah.
And now you gave them red and pink, and you asked how different they are. He said 5. Now you gave him red and blue and asked him how different they are. He said 5. Now you ask him blue and pink. How different are they? And he doesn't say 10. He says 7. But if you ask him a square and a rectangle, red and blue, then he said 10. That's correct. So there are some things which are seen holistically, some things which are not seen holistically. That's the point of Gardner. Okay. And the next thing is there are emergent features. Okay? Like there is something here and something there, and then it moves. Or there's something moving and then it moves faster. So you have position, from position you get motion, from change of motion you get acceleration. So there are new features which come out of old ones. And we see those new ones, and those new ones are separate, are independent of what they derive from. They're emergent features. Like we said, a melody rather than notes. Okay? In fact, we often have a precedence to perceive the compound rather than individual. If you ask someone, you play some music, and you ask someone, what was the music you just heard, you can say the melody. Now, with which notes was it? No. I mean, with perfect pitch, can't. And then Navon from Haifa came up with this thing. What letter do you see here? Okay. And the answer is usually T. It depends, of course, on the size of things, etc. But there's a forest before the trees. Forest before the trees. And then there are other places where you can combine things. If you have to find the line with the odd orientation, and say is it right, left, or that, it's faster to do this one. Well, it sounds obvious because this one is a completely different shape. But all we did was add this L shape to the line. So the whole, the gist, the holistic thing, the gestalt, is faster than its elements. We do not see this triangle because we see three lines. Because if we did, then the difference between them would be the same as the difference between them, right? But this is seen faster than that. That's not true in this case. It is true here. This is more different than this is from the others. And it's not true here. Okay? So again, it's emergent features that are coming out in a scene. Okay? Or forest before the trees. How quickly do we see if we're asked are these shapes with closure or without? With closure or without closure? Okay. Then, but there's also the global one has closure. Okay, how do they interact? And you can more easily see the global one than the local one. So that these two are more similar than these two. And these two. Okay. These have closure and these don't. Even though it's these two which have closure. Or orientation. This is vertical, this is horizontal. But here, what about here? This is vertical or horizontal? Vertical. This is horizontal. Now look at it. Holistic. The extreme came with a Gibson. James Gibson and Eleanor Gibson. And what they said was that there is something what they call direct perception. Now this is very complicated and it's hard to explain, mainly because everybody objects to it. And nobody talks about it anymore. Okay? In fact, Shimon Ullman wrote a paper in what's called Brain and Behavior Sciences, which is a journal where you write a paper and then people respond to it. And they publish the paper together, the responses. And he wrote against direct perception. Okay? And there were tens of responses to it. There's a book called Against Direct Perception. Because what they're trying to say is, more or less, you don't need computation. Now, we all know you need computation. But no, they said you don't need computation. What you pick up from the world is the important features, the important informational features of what's happening out there. And you do do that directly without going through a whole process. Direct cessation, direct information pickup of the relevant information. Seeing an environment by an observer in that environment is direct and not mediated by visual sensation or sense data, but rather based on the pickup of information. It's not that our retinas pick up where there's light and there's dark, and out of that we build objects. We pick up that they're objects. I know that this is a classroom full of people, not because there's green there and red there and blue there and so on. Okay? It's direct. Now, one of the things that has to be included is proprioception, that I know that I'm in this room. If I wasn't in this room, then I would perceive something totally different. So there's a relationship between perceiving myself and perceiving the room. And as I walk around and things change, do things change as I walk around? They're not changing at all. But on my retina it's changing. So why do I see you as stable? Why don't I see you as moving? Okay? Because what I pick up is directly the important information that this is a classroom full of people. If I would pick up points of light and dark, then everything would change as I walked around. But it doesn't change. Not light sense, but an array of light perceived. It's the whole array that's perceived. It's what they call ecological optics. It's the optics which are there, not physical optics, but ecological optics. It's what helps us perceive the world. The ambient optic array, light feels flow. Okay? When I walk, this whole thing moves. Okay? Then I know that I'm walking. I don't know that that's moving, it's not moving. I'm walking, that's how I know. Connected sets of observation points. At each point where I stand, I see a different world. And as I move, those worlds change. But that connection and the way it changes tells me more about the world. It doesn't change the world, it tells me about the world. Not seeing now and remembering what was before, but the act of seeing includes exploration and a family of different points of observation over time. The basic sense is not one flash and then a minute later another flash. Not one fixation of the eyes and then as I move my eyes a new fixation, then I remember what the old one is and compare it to the new one. No, it's all this is integrated together into one gist. Invariance versus changes with changing points of observation or illumination. For example, contours are invariant with illumination changes. Just because I lower the light, so to align them, the same contours are there. One second. Textures don't change, they're invariant with observation points. Contour, closed or open, is always invariant. The difference between a circle and a C doesn't change if I turn on the light, and it doesn't change if I look at it from a different angle. Open and closed. These are invariants which remain, and therefore I know something about the things I'm looking at by seeing that they're invariant. Even changes are local. When I do see a change, it changes in a local way. As I move along, and things change, and he hides him, and so on, it changes in a local way. Depth is nothing but breadth seen from the side. There's nothing special about depth, say Krishna. Just because things are further away and closer, how do I know that? In the same way that I see when things are lateral. Okay. You had a question?
Why is that not computation? All of these things sound very computational.
Yeah. It's probably my fault in the way I'm saying it. They found ways of expressing these things without using the word figuring out or computation or whatever. They said this is directly there. Now, one of the problems is, what does direct mean? Okay. And this is a real problem, okay? We think of direct as the photoreceptors receiving light. That's direct. Everything after that is not direct. Or, in terms of lesson, what the retina tells the brain. What the frog's eye tells the brain. So then we're including even the processing of the eye as you include everything, then what have you said? And you said nothing. Okay? So there's a problem here. The more they include in the direct, the more stages, then they're saying, okay, but then that's computation.
I think it depends why, how it's done. Well, just, you can't say, well,
Right.
you have to say like how.
Yeah. So, yes. They try to avoid saying that it's a computation. They're trying to say that it happens directly, automatically, without thinking about it. Just like motion can be picked up by a speedometer, or acceleration by our vestibular system is picked up without first knowing any location. So you can use location to know velocity and use change of velocity to know acceleration, or there's some way of knowing acceleration directly. That's what an accelerometer does. That's what our vestibular system does. We know about acceleration without knowing position. The vestibular system knows nothing about the position of the head. It only knows if it's accelerating. So in the same way they say there are things which we derive directly. What does that mean and how is it done? Okay? It's a question. I mean, there are good reasons why people don't like direct perception. Okay? But it's important to know that there is something here, okay? And okay. They ask the following questions, which other people ask and they don't ask. Why is the world upright when the retina is upside down? Why is the world stationary when the retina is constantly moving? We have constant saccades, and yet the world seems stationary. Why? Why is the world unbounded? Why do I don't see an end to the world over here? I can't see what's further. Why doesn't it seem like there's an end to the world? Why are size, form, color constant in the changing retinal views? And their answer is we do not see our retinal images. We see the environment. That's why it's not changing. That's why it's not bounded. That's why it's not upside down.
In order to see the environment, there has to be some process.
Yeah, but they're saying, at this point what they're saying is, you need to answer this question. Why don't we see an end to the world? Why doesn't the world look like it's got an end? It goes from here to there, and that's it. Beyond that, I don't see. It should seem like it's the end. When I look through a window, I see the frame of the window.
It's like the blind spot. Why don't you see the...
Why don't you see the blind spot? So it's a fuzzy end. So why don't I see a fuzzy end? If I took a window and put at the edges a fuzzy layer, then I would see a fuzzy end. But when I look at the world, I don't see it. Because, yes, you can answer. There are lots of answers. My experience is such that I know that if I turn my head around, the world continues. And maybe I've already done that until I remember what was there. Do I really remember what was there? Their questions are good. There are answers to each of these questions, but the answers are not as good as the questions.
What does it say with their answer though?
Their answer is we don't see the retina. It's not that the retina has a picture of the world when we look at the picture. Rather, we're looking at the world. And the world is not changing. The world is not upside down. We're seeing signs and elements and symbols about the world, and that's what we're getting. Just because the symbols are upside down doesn't make any difference. One of the things that we see are affordances. What can we do with the thing that we see? Is this a cup of coffee that I can drink? Do I see drinkability? Do I see drawability? Do I see tool ability? Those are the things that they say we also see. We don't derive them. We see them immediately. When I see my grandmother, I immediately go over and give her a kiss. Without thinking. It's part of it. So what is transduction? By transduction we mean changing energy in the world, just a picture inside, a representation inside. Is it only the photoreceptor response, or the retinal, or the whole brain? And that's what we said before. It's a question. If you go too much, then you've lost meaning for this whole thing. What is there? Patterns over space and time. Perceiving change. I mean, one of those things that putting time in is very important. Okay, and certainly the computationalists have to do the same thing. Somehow say that we're not computing what is now in the picture, and then we're doing a whole thing all over again the next second, and then we combine them. But rather, how things are moving and changing with time. Perceiving change is not dependent on memory, but like perceiving velocity and acceleration without comparing position at different times. Okay? It's a very difficult theory. Okay. The last theory I want to talk about is one of Mirava Chisar and myself. Okay? And in a way, it combines the 2 points of view by saying that direct is different than indirect. Direct is different than some sort of computed. Certainly direct is different than conscious, but it requires computation anyway. What we said was that there is what we call the reverse hierarchy theory, was that we get implicitly, quickly, the gist of the scene, and only with scrutiny the details of the scene. I have this again later. So reverse hierarchy theory provides a theoretical framework in terms of brain size and mechanisms for division that is found between two modes of conscious perception. The first is rapid vision at a glance. What happens when you close your eyes, you open them, what do you see? And slower visual scrutiny as you look around and study what you see. And this is done on the basis of the hierarchy of cortical areas, which have vision information processing and the massive feedback that's found amongst them. Nobody told you, but for every area of the brain that gives information to the next area, like LGN to V1, or V1 to V2, there's as massive a feedback from V2 to V1, or from V1 to the LGN. Okay. What's it doing? And all those models that we had of Google and Weasel and David Marr, who talked only about feed-forward processing, what is the feedback for? So here we're going to give use for these things. What we're saying is that the feed-forward connections from simple to complex to areas where we have shapes, basic shapes, to areas where we have cells which respond to faces, to grandmothers, to cars, to houses, and so on. The feed-forward first run is implicit. That means without conscious awareness. And it's feed-forward. But then, once we got to the top, and we become consciously aware of what is out there, the gist of the scene, we then have feedback, which goes to find the details. So it finds the detailed position of the things, it finds the detailed orientation, whatever details are useful for us at that point. So the lower areas respond to simple geometric forms in specific locations. The higher areas represent complex objects and categories, generalizing over position, luminance, orientation, and viewpoint. And the reverse hierarchy theory says the following. Processing begins with sensations of specific detailed information, which is analyzed and integrated into complex categories. Okay? Just like Google and Weasel. Except, not all neural activity is available to consciousness. And conscious perception, explicit action-promoting remembered perception begins with a generalized percept at the top and later dips to find specific details of the current view. Okay? Yeah.
Why do you need feedback for this?
What I'm saying is that consciousness goes back. Oh, why do you need feedback? I'm sorry, to direct. What you want to do is you say, I see a tree over there. Now I want to know the details of the tree. exactly the color, how big, how bright, and so on. So I go back to, and now I know where to go back to. I know what level of detail to go back to. So I go back, once I've seen the tree, I then can go back and find the detail. So I use the higher level as a guide to where to go back.
So, in your tree example, all of the light that's reflecting off the mirror, or whatever my photoreceptors are able to take in, they take in. So, what you're positing, I think, is that first you have a process where you have sort of a maybe a compressed input version where you have less detailed information, and then you sort of make sense of that, and then you go back to find the details. But
Right.
but physically that doesn't seem to be what's happening. In other words, physically all of the light is hitting your retina, and all of that information.
That's exactly the point that we're making. That physically, if you look at the hierarchy, what we get first are the details. So we ought to know the details before we know it's a tree. We should know the leaves. But we don't. What we know first is the tree. And so we're saying that is because we build up this gist of the scene. And we're only consciously aware of the gist, not of the detail, at first. Then we can go back and get it.
But so are we going back to the
The
environment?
brain has it, but we don't have it.
So we're not going back to the environment and getting the details
No,
from the
we're
environment.
getting it from
We're
the
going
back to the brain, lower areas of
lower
the brain.
level.
Yeah, you can close your eyes at that point. Okay?
When you mean explicit, what do you mean? That
Explicit, I mean conscious. Consciously aware. You know, it's difficult to talk about consciousness because nobody knows what it is. And I've been to conferences of the Association for the Scientific Study of Consciousness. And it's a lot of nonsense. So, I don't mean that we can't study consciousness. We're going to have to study consciousness. But a good first step is to know what we're consciously aware of, what we consciously perceive, rather than what we don't consciously perceive. Okay? Because I think in order to study consciousness, we have to study the subconscious. We have to study also what we're not consciously aware of. Okay. So therefore, what we're saying is that early explicit perception is a post-processing, high cortical level generalized view of the integrated complex categories and concepts using spread attention. And later conscious perception is a return to lower cortical areas with specific details of information available there using focused attention. Okay. So I'm going to give you an example. I'm going to show you two pictures, and I want you very, very quickly to tell me the difference between these two pictures. Okay? What's the difference? A dog and a tree or whatever. Right? But what else? There's window, the steps, one step or three steps, square window, round window, position of this window, position of the chimney. So we get the gist of the scene first. Everybody responds immediately. There's a dog and there's a tree. Okay? And the other thing, if you happen to be looking exactly at that point, maybe you see it. So we get the gist first. How come? I mean, we all know that it comes into the retina. The retina sees points of light and dark. And B1 sees lines. So why don't we get that first? And don't we have to use that to find out if it was a dog or a tree? Yes, but we're not consciously aware. So what we're saying is that this direct perception is not something to be thrown out. It's that it's something which involves the processing, but implicit, unconscious processing. And then we can go back and use the other. Yeah.
I'm not sure that this example is a good demonstration of your theory because, at least to me, I think that the issue in this particular case is a scanning and segmentation issue. In other words, the dog and the tree in these pictures happen to be separate from everything else in the picture. And I don't think that if I were to look at it and then close my eyes, I would then be able to find all that information already in my brain. I think that the process of scanning
Have you seen this? Have you seen this? Have you seen this before, you guys?
This picture.
What? Oh. I can't see it. Why can't you see it? You have it? Sorry. Let's try and get it. That happens sometimes. Then I should use the other one. Let's try this. Yeah. Have you seen this before? If you haven't seen it before, then tell me what is the difference between picture 1, 2, 1, 2. And don't tell me, raise your hand when you see the difference. 1, 2,